Welcome to the seminar series. Myself Supraja and I'm doing my second year of Masters in Central University of Tamil Nadu in Microbiology Department. And today we are going to see about the topic bioleaching. So basically what is bioleaching? So bioleaching is a method of employing microorganisms to extract metal from their ores and solid metallic waste. So the basic thing about this bioleaching is we are employing microorganisms to extract metals. So the metals can be extracted in various forms, like various types. So they are extracted from their ores and their solid metallic waste by employing microorganisms. So moving on to the next topic, we are going to see what is basically bioleaching. So bioleaching is also called as biomining or microbial bioleaching. So it is a biotechnological process that employs microorganisms to extract metals from their ores and their solid metallic waste. So various types of microorganisms are used in their extraction process, and we are going to see one by one. And so now we are going to see about the types of bioleaching. So what are the types of bioleaching? So the bioleaching can be categorized into three types: indirect bioleaching, direct bioleaching, in situ bioleaching, and slope bioleaching. So indirect bioleaching is a process of indirect bacterial leaching, which is non-contact bioleaching. So the surface of the bioleach, that means the surface of the slopes or the ores will not be in direct contact with the microbial organisms that we are giving to them. And direct bioleaching. So direct bioleaching is nothing but the contact bioleaching. So contact bioleaching is nothing but the direct contact between the bacterial and the microbial ores. And in situ bioleaching. In situ bioleaching is nothing but the in situ method. Like we are not going to change the places, like we are going to stay in the same place. In situ itself, we are going to leach the ores. The microorganisms are brought to that place in the site and then they are leached. And slope leaching is a process of leaching via a slope. So we are going to see that one by one. So now we see about slope leaching. So slope leaching is a hydrometallurgical extraction method in which low-grade ore is stacked on a natural hillside or a man-made slope and irrigated with a leaching solution. So this solution flows downwards by gravity, dissolving valuable metals. So now we'll see how it works. So ore is crushed. Usually ore is like a type of a coarse-sized metal. So the ore is crushed and it is placed on a prepared slope lined with a impermeable material. So after this, there is a leach solution, which is like an acidic solution as well as an alkaline solution, is sprayed or trickled from the top. So this because of the gravity causes the solution to percolate downwards, dissolving the target metals. That is the copper, gold, uranium, or any kind of a metal. Because of the gravity, it percolates downwards. So by this, the pregnant leach solution, the PLS, is collected at the bottom, and then the metals are recovered using a process called as solvent extraction or exchange or precipitation. That's what I'm saying here. The slide says the fine ground ore is ground and placed in a slope, and then continuous sprinkling of water containing the bacteria is then done, and then as the solution trickles down towards the ore, it gathers at the bottom and rich with the dissolved metals. After that, the low grade ores and the concentrated metals are economically taken back. So now we'll see about heap leaching. So heap leaching is a similar process kind of a slope leaching, but it involves the ore being arranged in large heaps known as the leach heaps. An aqueous solution containing the thiobacillus is sprayed over these heaps. The solution percolates towards the heap, dissolving metals as it goes. So metal laden solution is collected at the heap's base and then it is processed to extract the metals and sustain the bacterial culture in oxidation form. So now we'll see how it works. So the first, the ore is crushed. So ore is crushed to a uniform size to improve permeability. And then the heap is constructed. A heap like kind of a thing is constructed and the crushed ore is stacked on a lined leach pad. It is called as impermeable HDPE or a clay liner. And after that, irrigation process. In the irrigation process, the leach solution, which is more like a dilute sulfuric acid or cyanide or any alkaline solution, is sprayed from the drip emitters or sprinklers. After that, a percolation step. In the percolation step, the solution trickles through the heap by gravity, dissolving the target metals. After that, the collection. The metal-rich pregnant leach solution drains to collection pipes at the base. After the process, it is the recovery where the metals are extracted by the solvent extraction, electrowinning XEW, activated carbon adsorption, ion exchange, or precipitation, and then it is recycled. The balanced solution is reconditioned and recycled back to the heap. That is how the ore is collected at the end of this process. This is called as heap leaching. Next, we'll see about the in situ leaching. So, in situ leaching does not require the ore to be removed from its natural location. The ore will be placed at its natural location and we are going to hydrolyze them there itself. So, instead, after surface blasting to increase permeability, water containing thiobacillus is pumped through drilled passages within the ore body. So, this solution slowly seeps through the rock, the hydrolyzed metal. So, this seeps through the rock, dissolving the metals. The metal-rich solution is then collected from the base of the ore body and the water is reused after the bacterial culture has been regenerated. So we'll see how it is done. So the well fields are installed. So the injection well fields and the recovering pumps are all drilled into the ore deposit. And after that, the injection of the lead solution, the acidic solution for copper or alkaline carbonate solution for uranium and other lixiviants for the, it depends on the type of the mineral. For each type of mineral and each type of solution is used. And then the leaching underground. The solution moves through the ore bearing formation, dissolving the target metal in situ. And the recovery of the pregnant solution. The metal rich pregnant rich solution is pumped through the surface through recovery wells. And after that, the metal is extracted. The metals are recovered using ion exchange or solvent extraction or electrotwinning or precipitation. And then after that, the recycling of the leaching solution. So after that, the leaching solution is being recycled, it is being treated barren solution is then re-injected into the wells. Likewise, the solution is being recollected back through the mineral rich body and the bacterial culture has been regenerated and it can be used for other purposes to enrich the other sites. Like you can take other sites for example. Now we'll see about the direct bacterial leaching, which is a contact leaching. So in this method, the microbes directly interact with the mineral surface. With the physical contact between the bacterial cells and the mineral sulfide surface occurs. And in this example, thiobacillus ferroxidans catalyze the oxidation of the minerals. The bacteria tend to attach to the specific sites on the mineral surface and they reduce it. So the mechanism by which it works is the microorganisms, mainly the acidophilic and the chemolithotrophs, obtain energy by oxidizing the reduced sulfur and the iron components present in the metal ores. These biochemical reactions causes the breakdown of the metal sulfide bonds and the release of the metal ions into the solution. This causes a natural acidification that enhances the mineral dissolution. So the key microorganisms that are basically used in the direct bacterial leaching are Acidothiobacillus ferroxidans, Acidothiobacillus thioxidans, Leptospirem ferroxidans, and Sulfobacillus. So this direct oxidation can also be applicable for other non-iron metal sulfides such as chalcolite, spherulite, which are zinc sulfide and the copper sulfide, which is like hard to extract. So the non-iron metal sulfides can be hard to extract, and by using this bacterial leaching, which is a direct contact of the microbial substances on the mineral surface, they can be extracted easily. So now we'll see about the indirect bacterial leaching, which is a non-contact leaching. So in this process, the microbes do not directly interact with the ores. Instead, they produce strong oxidizing agents such as ferric ions or sulfuric acids. And these agents dissolve into the ores to extract the metals. So the microbes generate Fe3 plus and H2SO4 ions in the surrounding solutions. And these compounds react chemically with these metal sulfides to release these metal ions. So the microbes remain in the solution, no direct attachment to the O surface. The microbial role is done is they oxidize the ferric ions into ferrous ions and they oxidize sulfur compounds like H2SO4 and etc. And the chemical role that they are doing is the ferric ion oxidizes the sulfide minerals and the H2SO4 dissolves the mineral matrix and maintains the low pH. The key microorganisms that are used here are Acidothiobacillus ferroxidensis, Acidothiobacillus thioxidensis, and various other perpetuating organisms that leach the process. So ferrous ion produced in this reaction can be reoxidized to the ferric ion by Thiobacillus ferroxidans or L-thioferroxidans, and thus the leaching process continues. By the presence of this thiobacillus, we can extract the metal ions and the ores by non-contact. So the bacteria will not be in contact with this ores and thus we can extract the metal. So now we are going to see the differences between the direct and the indirect bioleaching. So the direct bioleaching, in which the microorganisms directly interact with the mineral substrate where their enzymatic activity catalyzes the oxidation of the metal. Whereas in indirect bioleaching, we can see the microorganisms generate highly effective oxidizing agents that oxidize these metal sulfides. So what did we see in direct bioleaching? The microorganisms have direct physical contact with the mineral substrate, and their enzymatic activity catalyzes the oxidation of the metal. And in indirect bioleaching, the microorganisms generate some kind of effective oxidizing agents that can oxidize the metal sulfates. The microbial action involved is the microorganisms directly attaches to the metal sulfide ores, and in indirect bioleaching, an organism oxidizes ferrous to ferric ions, which reacts with the metal sulfates, which we saw in the previous slides. And the application that I used are in the direct extraction of the metals from sulfide ores, and in the direct extraction of the like in situ leaching and all, the direct extraction is being done. So here and all, we need not transfer the metal ions from one place to another. Instead, by spraying all the microorganisms in that place, the metal ion can be extracted easily. And this indirect bioleaching is used in the form of this heap leaching and all. In heap leaching and all, that is a heap of microorganisms, heap of ore present, in which we are going to spray the microorganisms on top of them, and the microorganisms produce certain amount of effective oxidizing agents. And these oxidizing agents, what will they do? They will produce certain chemicals, and these chemicals will leach the ores, and they help us to extract these metals. So these are the basic differences between the direct bioleaching as well as the indirect bioleaching. So in this figure, we see about the mechanism or the types of the bioleaching. I've already told you about the types of bioleaching. It is indirect bioleaching, contact bioleaching, and cooperative leaching. So in indirect leaching, the microorganisms produce certain kind of chemicals or oxidative agents that can reduce the ores into their metal ions. And in contact bioleaching, the microorganisms are in direct contact with the metal ions or ores, and they feed on the ores, and then they release the extracted metal ions. And in cooperative leaching, we have the contact leaching bacteria as well as a non-contact leaching bacteria, and these suspended bacteria feed on the released species, that is bacteria that are enabling the indirect leaching, and then they together form the leaching process. So these are the basic types and the mechanisms of bioleaching. The metal sulfide ore is converted into the normal metal extracted that we are using for our day-to-day purposes. So now we'll see about the factors affecting the bioleaching. So what are the factors that are basically affecting the bioleaching process? The choice of bacteria. The bacteria must survive and thrive in extreme conditions such as high temperatures, acidic environments, and elevated concentration of high metals. So we have to choose such kind of a bacteria like acidobacillus, thiobacillus, acidobacillus thioxidans, acidobacillus peroxidans, like these kind of an extremophilic bacteria that can thrive in extreme conditions and extreme temperatures, so that they can be used to extract these metals. And the pH and temperature should be checked. So the pH should be around 2 to 2.5, and the temperature should be around 28 degree to 30 degree, and that are crucial for maximizing the microbial activity and metal solubilization. So the bacteria to work optimally and to work in a better condition, we are going to give them a optimal pH and an optimal temperature, which should be around 2 to 2.5 for the pH, and the temperature should be around 28 to 30 degree. And the population density of the microorganisms, the higher the bacterial population, the higher the leaching rate. So the more amount of the bacterial population that we are going to spray on the leach, the more amount of the metal extracts that we are going to get. The oxidation reaction compositions, the low oxidation levels are necessary for achieving the fastest leaching rates. If there is a more oxidation level in the bacteria are obviously gonna lose their capacity and lose their optimal working capacity. So for that, we are going to use them as low oxidation levels, and they can give the fastest leaching rates. And metal tolerance of these microorganisms. The use of tolerant or resistant organisms avoids inhibition and ensures effective metal extraction. So the bacteria, that is particularly thiobacillus, tolerate high levels of heavy metals and can be used as a best option. The mineral substrate. High carbonate content present in the environment can increase the pH and inhibit bacterial activity. So the high carbonate content could be reduced, and then the requiring amount of acid addition and to maintain the low pH should be done. And the surface area of the ore or the particle size. So higher the surface area, the lesser the ore particle size. So likewise, we have to choose the higher the bacterial oxidation due to the increased area for bacterial action. So the more the surface area of the like the ore particle. So the particle or surface area should be less. So we can get the lesser amount of the ore particle size, and then the oxidation process could be done faster. And adding a certain amount of catalyst. Certain amount of catalyst like ions, silver ions, graphene, and various other catalysts could enhance the performance of these bacteria. So these are the various factors which are affecting the bioleaching processes. What are the basic factors? Choice of bacteria, pH and temperature, population density of the microorganism, oxidation reaction compositions, metal tolerance of the microorganisms, mineral substrate, surface area of the ore, as well as the catalyst. The applications of bioleaching. So the applications of bioleaching are environmental stabilization. Bioleaching stabilizes the sulfate toxins, minimizing the environmental impact. In the emission reduction, it eliminates harmful sulfur dioxide that are emitted and associating with the traditional smelting processes. It can destroy the health. So by using bioleaching, we can improve health as well as environmental outcomes. Cost-effective extraction. So the bioleaching is more of a cost-efficient method compared to smelting, particularly for the low-grade iron ores. And the optimized recovery process. So it can allow for an optimized recovery process for the involvement of the extraction of the metals. So these are the various applications of bioleaching, which also includes the recovery of metals and the bio-oxidation of refractory gold ores to improve gold extraction, and the extraction of metals from the mining waste, tailings or slags, and the bioremediation of the heavy metal contaminated soils and waters, and the recovery of the valuable metals from the electronic waste, the extraction of rare and strategic metals like lithium, manganese. These are all extracted from the ores and waste, and these can be extracted easily. And the breakdown of sulfide minerals to reduce acid mine drainage formation, and they are used in the industrial biohydrometallurgy processes, and they are used in the removal of impurities to upgrade one ore quality, and they are also used in the recovery of metals in the industrial catalysis. So these are the basic applications of the bioleaching. So these are the references which are used for taking this bioleaching process. Thank you.